This is the second lecture that is part of the cardiovascular system and circulatory system. Our blood has markers on the red blood cells that determine our blood groups or blood type. They are expressed codominantly. You inherit one marker from your mother and one from your father and express both at the same time. Those with blood type A form antibodies against the marker they don't have, so they will have anti-B antibodies. A person with blood type A will either have 2 genes for A or 1 for A and 1 for O. They will be type A since O is actually for no marker. Type B makes anti-A antibodies and has both B genes or 1 for B and 1 for O. Type AB has both genes but does not make antibodies towards either. The O has no markers on either gene but makes antibodies towards both markers. There are several links in Angel that you'll be able to look at to practice the concept that goes through with the different blood groups. The Rh group is another marker identified in the rhesus monkey. The trait is dominant in its expression, so if one parent is positive for the gene and one is negative, the baby will be positive. This is not a problem unless the mother is negative and the father is positive. There is no problem during the first pregnancy generally, but in subsequent pregnancies the mother's body has developed antibody to the marker she does not have, and her immune system will attack the fetus, often terminating the pregnancy. Now, an Rh-negative mom is given an injection during her pregnancies to prevent her immune system from forming the memory cells to attack a future fetus. Here we have a blood group animation. Many different antigens or cell surface molecules are associated with human erythrocytes. The most important are the antigens of the ABO blood group and the Rh group. The ABO blood group is based on antigen A and antigen B. Blood types are determined as followed. Certain antibodies in the plasma are synthesized some months after birth. Type AB has no antibodies. The universal recipient can theoretically receive any blood type, but it is best to give this individual type AB. Type O has anti-A and anti-B. The universal donor can only receive type O, but can give blood to all other types. It is best to give this blood as packed cells because of the presence of both anti-A and anti-B in the donor plasma. Type A has anti-B. It is best to give these individuals type A blood, but they can also receive type O. Type B has anti-A. It is best to give these individuals type B blood, but they can also receive type O. In blood transfusions, it is critical that an individual's red blood cells not come into contact with antibodies against his or her own antigens. Here we look at the Rh antigens. If the woman in the second example were exposed to some of her child's Rh-negative blood during pregnancy or delivery, her body would respond by producing antibodies that would attack the red blood cells of a second fetus if that fetus were also Rh positive. RhoGAM, an anti-Rh antibody, can be given to the Rh-negative mother during late pregnancy or shortly after delivery. RhoGAM binds the Rh positive fetal cells and shields any Rh positive fetal cells that might come into contact with the mother's cells. The mother, so treated, does not produce anti-Rh antibody. And you'll be able to practice the little quiz by having access to this outside of this presentation. The heart has a triangle shape and it sits in the body tilted to point down and slightly towards the left side. The base is the flat top side, while the apex is the bottom pointy side on the left. The heart has 2 lower exit chambers called ventricles and 2 upper entry chambers called atria. The heart has a series of coverings like any other organ. The pericardium is the membrane surrounding the heart cavity and helps hold the heart in place. The outer portion is the fibrous pericardium and is tougher and more protective. The inner portion is the serous pericardium and is thinner and more delicate. There is another system for naming membranes around organs. The parietal layer forms the outer membrane and sac around the organ. In the heart, this is the pericardium. The visceral layer is the inner membrane that is adhered directly to the organ. In the heart, this is the epicardium. The epicardium is the visceral layer adhered directly to the heart itself. Between the epicardium and pericardium is a space called the pericardial cavity. This space contains pericardial fluid to reduce friction. The space between the lungs, which contains the heart and its vessels, is the mediastinum. The outermost layer of the heart wall is the epicardium. Deep to that is myocardium, or muscle tissue. The myocardium is cardiac muscle with intercalated discs, or thickenings in the sarcolemma. The gap junctions are the area of the intercalated disc that allows rapid conduction of the action potential. The innermost layer is the endocardium. This is made of simple squamous cells and is found lining the chambers forming valves and tendons and is continuous with the endothelium of the blood vessels. The upper entry chambers are the atria. Blood enters the right atria from the body and is deoxygenated. Blood enters the left atria from the lungs and carries oxygen. The ventricles are the lower exit chambers. The right ventricle pushes deoxygenated blood to the lungs, and the left ventricle pushes oxygenated blood to the body. The inner atrial septum divides the right and left atria. The interventricular septum divides the right and left ventricles. A fetus does not acquire their oxygen from the lungs, so there is an opening called the foramen ovale in the interventricular septum, which allows blood to bypass the lungs. This hole closes at birth with the first breath. If it does not close, it is referred to as a patent foramen ovale. Here we're going to watch a little animation on the heart anatomy. This amazing organ has 4 chambers. Each one is separated from the other by cardiac muscle. Examine the illustration below. Can you see the valves opening and closing between the atria and the ventricles? The AV valves are between the atria and the ventricles on each side of the heart. On the right side, the AV valve is called the tricuspid. The left AV valve is called the bicuspid or mitral valve. The pulmonary semilunar valve is between the right ventricle and the pulmonary trunk, and the aortic semilunar valve is between the left ventricle and the aorta. The AV valves close to prevent any blood from flowing back into the atria when the ventricles are contracting. The tricuspid has 3 flexible cusps or flaps, and the bicuspid has 2. Chordae tendineae are attached to the papillary muscles. When these muscles contract, the white string-like chordae tendineae become taut. It is these chordae tendineae, together with the papillary muscles, that keep the valve shut when the ventricle contracts. The blue blood entering the heart from the body is oxygen poor. The red blood is oxygen rich and is returning to the heart from the lungs. And now you can go ahead and complete the rest of this on your own. You do have access to this tutorial outside of this presentation. The superior and inferior vena cava drains the deoxygenated blood from the body into the right atria. The coronary sinus drains the deoxygenated blood from the heart muscle into the right atria. The blood moves from the right atria into the right ventricle and exits the pulmonary trunk, still deoxygenated. This will split into the 2 pulmonary arteries traveling to the lungs, where the blood becomes oxygenated. It returns to the heart via the pulmonary veins and enters in the left atria. Next, it flows to the left ventricle and exits via the ascending aorta and carries oxygenated blood to the body. The heart has valves to keep the blood flowing in the proper direction and prevent backflow. The valves are made up of endocardium folded back on itself. The right AV valve or tricuspid valve is located between the right atria and right ventricle. It is called the AV, which means atrioventricular, and the tricuspid because it has 3 flaps. The left AV valve is also called the bicuspid valve for only having 2 flaps. It is also known as the mitral valve and is located between the left atrium and left ventricle. The AV valves have chordae tendineae, which are tendons made of extensions in the endocardium to prevent the valves from opening backwards. They connect to projections in the cardiac muscle called papillary muscle. The semilunar valves are half-moon shaped. The pulmonary or pulmonic semilunar valve is at the entrance to the pulmonic circulation, and the aortic semilunar valve is at the entrance to the systemic circulation. The coronary circulation is the blood flow to the heart muscle itself. There are 2 major arteries, the left and right coronary arteries that branch off the 2 sides of the heart. Each of these branches into 3 branches. The coronary sinus drains the deoxygenated blood from the heart. The more work the heart does, the more blood vessels and oxygen it needs. The blood vessels will respond by forming more blood vessels, and they will form anastomoses, or connections, between the branches of the arteries to provide alternate routes for blood flow. A person with more cardiovascular activity is less likely to have a blockage in the blood vessels of the heart or myocardial infarction. But in the event they do have one of these, the heart has better odds of surviving because there is an alternate route to keep blood flow going. The muscle contraction in the heart has a rhythm established by the pacemaker. The pacemaker is a special group of cells that generate an action potential in a rhythmic pattern to set the pace for the rest of the cardiac muscle. Without a pacemaker, the cardiac cells would all be contracting rhythmically on their own and not coordinated. The SA node is your primary pacemaker, and it is where cardiac excitation actually begins and sets the faster pace for the heart. This is located in the right atrial wall. The AV node is located in the inner atrial septum and slows the action potential started by the SA node. This is important to delay the ventricles to allow the atria to empty into the ventricles. The action potential continues to spread down through the AV bundle, or the bundle of His, in the interventricular septum. The right and left bundle branch split to carry the impulse around the apex of the heart. The Purkinje fibers will be used to rapidly conduct the action potential over the remainder of the ventricles. Relaxation begins at the end of the cardiac cycle. Here we have repolarization and a decrease in pressure that opens the AV valves to allow passive filling of the ventricles to about 75%. The remainder is filled by the contracting atria in systole. This is the contraction phase, where diastole is the relaxation phase. There are 2 heart sounds made by the valves closing. Lub is the first sound made by the AV valves closing. Dub is the second sound made by the semilunar valves closing. In this slide here, we will look at the electrocardiogram. EKG is just an old spelling of electrocardiogram, and the name has still stuck around. The P wave represents atrial depolarization. And you can see this going on right here. The time frames mentioned here are to give you a relative perspective of how long each of these phases lasts. However, if you speed the heart up or slow it down, these numbers would change. Next, we have the QRS complex here, which is the combination of these waves that represents the onset of ventricular depolarization. And finally, we have the T wave here that represents ventricular repolarization. Although we do also have repolarization of the atria, it occurs at about the same time frame you would have the QRS complex here with the ventricular depolarization.